Okay, so we're talking about the gene today and the structure of the gene. And if you recall back to our very early lessons, we introduced to you the concept of the cell theory, that the cell is the basic unit of life. All living things contain cells, and all cells arise from pre-existing cells. Those are some of the postulates of the cell theory. And so with that, we're extending that conversation today to look and see how cells get their function. Okay, so we know that all cells contain genetic information in the form of DNA. Okay, this DNA is organized into structural units that we call the gene. All right, these genes, as we saw on last week, and we discussed inheritance as far as Mendelian genetics are concerned, these genes are responsible for controlling the various features that we see about each other, and we use the term phenotype, right? So our genes determine those. So we're going to look today at the gene in more close detail, and the structure of the gene and how we go from the genetic information that's found in our DNA to all the way to a functioning protein product. And that protein product, therefore, those protein products, therefore, influence the function of our cells. Okay, so I'm going to share my screen with you and we will discuss the gene structure and function. Let's see. There we go. So gene structure and function. So just as an introduction, all living organisms have some common characteristics that we've talked about these since day one. You know, traits like replication, nutrition, growth, interaction with their environment. Okay, so an organism is composed of organs, right, which perform specific functions, right? Organs are made up of tissues, which are just aggregates of cells with similar functions. Okay, so when we're talking about the cell is the basic unit of life for all organisms. Okay, and the cell contain molecules that have fundamental functions for their life, right? And those molecules are what we study in the branch of science known as molecular biology. Okay, we're studying these molecules inside of the cell, particularly the protein. Okay, so there are two fundamental molecules that we need to be concerned with when we're interested in how does the cell function to sustain life, and those are DNA and protein. Okay, so DNA is made from these building blocks that we call nucleotides. Okay, so if you heard about this term nucleotide before, go ahead and write it down. Okay, there are two fundamental molecules inside of the cell that are responsible for its function. So we have nucleotides, right, which are the building blocks of DNA. Okay, specifically, we're talking about the pieces of DNA that are known as genes, and these genes encode proteins. And we sort of looked at the protein a little bit when we talked about the biological molecules. We talked about carbohydrates, lipids, proteins, and nucleic acids. Today, we're going to take a further dive and see how we actually go from, again, DNA all the way to proteins. All right. We said that our DNA was our blueprint for life. All right. So we're going to see how the information contained in our DNA, okay, how we have these genes, and how we end up with these functional products that we call proteins. Okay, so proteins are a very key component to everything that's related to life. Okay, it is the protein expression within a cell that is responsible for all of the biochemical activity that's taking place within the cell. Okay, so think with me. Your body has many different hundreds of different cell types, right? And all of these different cell types have different functions. So how is it then that these that the skin cell functions different than, say, a beta cell in your pancreas? How does you know, a sperm cell function differently than, say, a red blood cell? Okay, we're going to take a look at that today. And the short answer is due to the proteins that are expressed in those cells at any given moment. Okay, So let's look at DNA for a second. So we're talking about the two major molecules that are responsible for functioning of the cell. So we have DNA and then we have protein. All right. Really quickly, just to recap, we know that DNA is a very strong molecule that we find within the cell. It contains all of our genetic information, and it is arranged in a structure that we call a helix. Okay, structurally, we refer to DNA as a double helix. Okay, so we have two strands of nucleotides that wind around each other in this helical formation, kind of this spiral format. Okay, this is a double helix. Okay, these double helices assemble as we attach adjoining nucleotides together. All right, these nucleotides are held together by bases, two different types of bonds. Okay, we have hydrogen bonds and we have phosphodiester bonds. Okay, but so this is what it looks like from a 3D structural standpoint. We see this helical structure, but if we looked at it from sort of a 2D format, we just see two linear lines of nucleotides. Okay, that are joined together by these nitrogenous bases in the middle. So you should be familiar with the structure of DNA, and that again, this double helix is formed from two strands of nucleotides, okay, that are joined together in the middle by these nitrogen containing bases, and we call them nitrogenous bases, okay. These nitrogenous bases are held together, are linking these two strands of nucleotides together, okay. There are 4 nitrogenous bases that you should be familiar with: adenine, thymine, cytosine, and guanine. Go ahead and write those down. Okay, we talked about them a little bit back in an earlier chapter. Okay, so from a 3D standpoint, we can see this double helix, but if we're just looking at it from a linear standpoint, we see DNA as a double-stranded molecule where we have 2 strands of nucleotides in a linear sequence, and then these 2 strands are joined together by these nitrogen-containing bases, nitrogenous bases. So where are we going today? We're talking about this concept, the central dogma. Write this in your notes, okay? When you go out and read through your study module, you're going to read a lot more about the central dogma of molecular biology, okay? This is a very fundamental principle for, again, understanding how our cells function, okay? So we have the DNA, okay? We said DNA is the blueprint for life, okay? This is sort of our starting material or molecules, okay? This DNA goes through a process known as transcription. Write this word down, transcription. DNA undergoes a process known as transcription, and it is transformed into an intermediate molecule that we call RNA. Okay, this RNA then goes through another process that we'll call translation in order to produce protein. And so this central dogma represents the flow of information in all living things. How we start with the information in our DNA, okay, our genes, those sequences that we talked about, okay. All of the information in your DNA is used as a blueprint or a template for being able to produce final protein products that the cells need, okay? And so we're going to take a look to see how this happens with transcription and translation. So the process of going from DNA to RNA is called transcription. You should write that down. And then the process of translating this RNA into protein, we call that translation, okay? And you've probably heard about mRNA before, and that's because it serves as a message, okay? The information in this messenger RNA, we use it to translate or make proteins. So we're going to further explore this concept today of the central dogma. Okay. So we're going from DNA to RNA to protein. Let's start with DNA. What is the function of DNA? We've already established that the function of DNA is to carry, it carries the blueprint for life. What does that mean? Okay, it means that this DNA, these nucleotide sequences contain information, okay? And we read the information in DNA, okay, to during transcription to make this RNA, which is an intermediate molecule before we actually end up with our proteins, okay? It carries the blueprint of life. It is being duplicated whenever we make new cells. We looked at that in our unit on mitosis and meiosis and in the eukaryotic cell cycle. We saw how the synthesis of DNA, how new DNA is being made. But more importantly, as we're talking about cell function, DNA codes for protein, okay? And it is these proteins that are responsible for biological functions, okay? It is the protein expression profile of a cell that is responsible for biological function in that cell. So DNA is a very vital piece of this puzzle as we understand how our bodies function, okay? DNA serves as the instructions, if you will, okay? And so then I mentioned this process of transcription earlier. Write this down. Transcription is the process in which DNA is transcribed into RNA. Where does transcription take place? This process takes place inside the nucleus of the cell. Okay, so we already talked about some of those internal cellular structures back when we toured the cell, and the nucleus we know was one of those very prominent features that we could see and identify very easily. Okay, we know that it contained the DNA. Now we know that inside of the nucleus there's a process taking place known as transcription. And so what happens is DNA is going to transcribe or make a complementary strand of RNA that we'll call mRNA. Okay, so as you are following this step-by-step process that we'll call the central dogma, DNA is transcribed into RNA. Specifically, this RNA becomes a messenger. Okay, so we call it messenger RNA. Okay, this messenger RNA is basically some sequences of RNA that will later be used to produce a final protein product. We will read it through the genetic code, okay, to assemble a linear sequence of amino acids. And again, we talked about the amino acids already as the building blocks of proteins. We talked about those structures of the protein. So we have these linear sequences of amino acids that will be read from our messenger RNA. And then that will be used to, again, produce this final protein product. So let's keep walking to see how this happens. So again, let's walk back to our initial picture here. We're looking at what happens with this flow of information from DNA to protein. So we start with DNA and we have to make RNA first, okay? That process is known as transcription, all right? So we're going from DNA to RNA. All right, we're clear what the function of DNA is. It basically contains the blueprint or the instructions for creating RNA and ultimately protein. This process of transcription is a very important, first step here before we can get our proteins made. We have to create RNA, and this takes place in the nucleus. Okay. Transcription takes place in the nucleus and our DNA is transcribed into RNA. That's what you need to have in your notes.
Can you say that one more time while I'm writing?
Okay, so we're saying that the process of transcription is a process, biological process that's taking place inside the nucleus of the cell. Right. And what is happening during transcription is that DNA is being transcribed, okay, hence the word transcription, DNA is being, or so you can think of it as like a transformation, if you will, is being transformed or transcribed into a new molecule that we'll call messenger RNA.
Okay. All right.
Okay, and so this is kind of an intermediate step that's necessary for us to finally end up with protein. So it's kind of a two-step process. All right. And so visually, the flow of information looks like this. We have these genes, right, in our DNA. Okay, we have this double-stranded DNA, and we have genes here. And the process of transcription takes us from DNA, okay, to a messenger RNA transcript. A strand of messenger RNA is created. And we use a system known as the genetic code to basically decode the nucleotide sequences in our DNA into this messenger RNA. And it is these groups of 3 nucleotides that we call a codon in order to determine our amino acid sequence, okay, for translation, okay? But just stick with me for a second here and make sure that you're understanding this first step in the process, which is transcription. Before we can make protein, we have to make this intermediate molecule that we call messenger RNA. And the reason we call it a message is because right here, look at my cursor. We have some sequences of nucleotides here that we read in groups of 3, and we call it a codon. It's a set of 3 nucleotides that we read, and we're basically decoding this into amino acids. And it is these codons that will determine which amino acids are in our linear sequence that will be translated into protein. Okay, so we're going to keep walking through this, but make sure you write down the word codon. Okay, it's a group of 3 amino acids. I'm sorry, 3 nucleotides that will be translated into amino acids. So the second part of the story, or the second step in this process of creating proteins, is translation. Now, I said transcription takes place in the nucleus. So that DNA is transcribed into RNA. And so then that messenger RNA has to be translocated out of the nucleus. Okay, so it comes out of the nucleus, this mRNA transcript, out into the cytoplasm where we have ribosomes. That is the site of translation. Write that down. The second step in protein production is translation. It takes place on ribosomes. You know, ribosomes are found sort of free flowing out in the cytoplasm, and we can also find ribosomes associated with the rough endoplasmic reticulum. So, translation is carried out on ribosomes, and what's happening during translation? Very simply put, messenger RNA is being used as a template to create our proteins now. So this messenger RNA, as the name suggests, contains all of the necessary information for the synthesis of proteins. Okay, so the information that you should be grabbing right now is A, what is translation? Translation is the process in which messenger RNA is used to create a protein. Where does translation take place? It takes place in the cytoplasm on ribosomes. Your answer has to include ribosomes. Translation takes place on ribosomes. And so then how do we go from messenger RNA to actual protein? I'm going to tell you how. It's through the use of the genetic code. Okay, we use the genetic code to decode the information that's in our messenger RNA to make amino acids. And these amino acids are the building blocks of proteins. So I'll let you get that down for a second. Okay. So we're talking about translation now.
Sorry.
Okay. So these genes then, okay, we already talked about them being the, again, the sequence physically, they're just, you know, segments or sequences of DNA that are coding for our messenger RNA, all right? And so they, again, become the starting point for us to be able to produce these proteins through translation. So let's look at how this gene is actually organized. Okay, the structure of the gene is very simple, and you should be able to describe it and label, excuse me, the parts of the gene if you need to. Okay. So gene structure looks like this. Most genes consist of a short coding region that we'll call exons. Okay. Exons that are going to be spaced out or interrupted by longer non-coding regions that we'll call introns. Okay, so there's two parts of this gene structure that you should be familiar with. They are the coding sequences. These are the ones that we'll use to synthesize our protein, and then our non-coding regions will not code for proteins, and those are introns. Okay, so we have in red are exons, okay? And then these blue pieces are our introns. These are non-coding sequences that are there. So all of the information in our DNA is not used or translated into protein. And those sequences of DNA that are not coding for DNA, we call not coding for protein, we call them introns. And so then on the ends of each gene, right? So this represents, you know, theoretically one single gene here, okay? Remember we talked about genes last class that encode a trait, all right? So this would be a gene that we'll hypothetically say encodes a trait like, I think we used eye color last time, okay? And on the ends of each gene, we have some untranslated regions, again, that do not code for anything, okay? And so there are some modifications that will take place to this segment of DNA. So this is just one long piece of DNA, okay? And so to put it simply, some of the information in the DNA is going to be used to produce protein. That's going to be our red coding regions. And then some of the information in DNA is not used for protein production, and those are introns. And so as we move through this process of transcription and translation to get our final protein product, modifications are made to this gene, all right, to basically X out and remove these non-coding regions. Okay. And so as you're generally trying to form a picture in your head about the structure of the gene, genes must have some components, okay? They've got a start site. That start site is going to be where it actually starts to encode protein, okay? Those amino acid sequences. And then it has to have those exons, okay? The exons are our red coding regions. All right. And then there are obviously some points of control throughout this gene structure to control the start of transcription and translation and so forth. So how do we go from the information in our DNA to messenger RNA to protein? Okay. So once we have our DNA grouped into threes, into groups of three, we call those codons, okay? We have the designation of a start and stop codon at the beginning of each gene, okay? So the start and stop codons dictate where a functioning protein product will be made, okay? The messenger RNA, again, once it's synthesized in the nucleus, it's going to be translocated out of the nucleus into the cytoplasm. Okay, so I showed you this schematic earlier. Okay, we start with this double-stranded piece of DNA, and it's going to undergo a process known as transcription to produce our single-stranded messenger RNA. Okay, this messenger RNA now is traveled out to the nucleus and onto the ribosomes where translation will take place. And so what's happening during translation is that we have to decode the information in our messenger RNA, and we have to remove those non-coding regions through a process known as splicing. Write down the word splicing. mRNA splicing is the process that we use to excise or remove these non-coding regions, okay? As we just looked at the structure of the gene, and it's got two major components. We've got some coding regions that we'll call the exon, right? And we have our non-coding regions that are called introns, okay? So before we can make our final protein product, we've got to remove these introns. And we do that by splicing. Okay. And so RNA splicing is how we remove those non-coding regions to get a contiguous alignment of these coding regions, exons, of these exons, all right? And so then we'll take these sequences in our exon regions and decode them using the genetic code in order to align our amino acids, okay? What questions do we have about this? Okay. So let's recap here. We're saying the central dogma of molecular biology tells us that information flows from DNA, okay, the information that we use in our DNA, the information that our DNA contains, we use it to create messenger RNA, okay? This RNA serves as kind of an intermediate molecule that's necessary before we can actually produce our protein, okay? So this is how it flows. Genetic information is stored in our DNA, okay? Segments of DNA that encode proteins or other functional products, we call those genes, all right? So these gene sequences are going to be transcribed into messenger RNA, basically some intermediate molecules, okay? These messenger RNAs are then going to be translated into protein, okay? And these proteins are responsible for performing the functions or giving cells their function, their unique functions, okay? So you need to have a clear picture. You know, this is, you know, I've summarized this. It's a very intricate process, but I've summarized what happens, how it happens, and where, very briefly, how we use the information in DNA to make protein, okay? The two processes are transcription and translation, okay? We talked about how that messenger RNA molecule has to be modified a little bit to remove those non-coding regions so that we can get one contiguous gene structure, gene segment to code for our protein products of interest. Okay. So what are we saying here? We're saying that the genes are the basic physical and functional units of heredity. Okay, we talked about this on last class. The gene, okay, those segments of DNA are the actual units of heredity that's transferred from one generation to the next. All right, we already made clear that each gene is located at a very particular region on the chromosome, okay, the locus, right? We talked about that last week, and it has a specific ordered sequence of nucleotides. All right, and so it is those nucleotides that are going to be transcribed into RNA in order to make our final protein products. Okay, so genes are the physical and functional units of heredity that are responsible for biological activity. All right, so this is just another illustration of the structure of the gene. You should be familiar with this structure of the gene. All right, again, we have these red regions that are exons. The exons are the most important components when we're talking about protein synthesis, because they contain the sequences of nucleotides that will be translated into amino acids. All right. This is showing our RNA splicing again, how we have two different parts of the gene. We have our non-coding regions, which are introns, and we have our coding regions, the exons. And then this RNA transcript has to be modified through mRNA splicing in order to remove these non-coding regions, okay, so that we can have a linear continuous sequence of exons that will be used for translation through the genetic code. Okay, so again, what's happening during translation? During translation, we are using the mRNA template to now finally make that protein product. Okay, we said the translation occurs on ribosomes. Okay, and we read those groups of DNA, those 3 groups of DNA in codons, read them in groups of 3. Okay, those sequences of DNA, we read them in groups of 3 that we call codons, and each one of those codons code for an amino acid. And so it is this linear assembly of amino acid that gives us our protein, or that polypeptide sequence. Okay, so how do we go from DNA sequences to amino acids? We use the genetic code. Write down this term, genetic code. The genetic code describes how nucleotide sequences in our DNA is converted to protein. We use the genetic code. So we read those nucleotide sequences in groups of 3, and that creates a codon. And each of those 3 groups of nucleotide sequences codes for a specific amino acid. And it is these amino acids that are going to finally become our polypeptide or protein sequence. So the amino acid is the building blocks of protein. We know that there are 20 amino acids that exist in nature. Okay, there are various codons, okay, nucleotide sequences that code for a particular amino acid. All right, there are 20 amino acids found in nature, but the various combinations of bases can form up to 64 different codons. Okay, so what's the end of the story? These proteins. Okay, the proteins are special cellular components that special cellular components called ribosomes uses the genetic code to translate nucleotides of messenger RNA into an amino acid sequence of a protein. If you remember from our previous lecture, we talked about proteins and how amino acids are the building blocks and how these linear sequences of amino acids make up that primary structure of proteins. That's what we have here. So there are 20 different amino acids that exist, and proteins are being created by linking together amino acids in a linear fashion to form a polypeptide chain. I'll give you a minute to write that down. Okay, so we're looking again at this concept of the central dogma, how we go from DNA to RNA to proteins. All right, these proteins are what you should understand is that they are now the functional units of cells. Okay, proteins are responsible for giving cells their specialized function. Okay, these proteins are created by translating the information from that messenger RNA into amino acids. Okay, and so it is the amino acid sequences that make up our primary protein structure. Okay, that's what that polypeptide chain is. It's just a linear sequence of amino acids. Okay, and so, you know, we spoke previously then about how these protein polypeptide chains can fold and assemble themselves into various 3D structures that can influence their specific functions. Okay, so we talked about the primary, secondary, tertiary, and quaternary structures of protein previously. Okay, and it is those functions, those structures that dictate the function of the protein. Okay, and so, you know, just to recap again, proteins have 4 levels of structure, and there is a direct relationship between the structure of the protein and its function. We already looked at the many functions of proteins, that they can have some functions in structure in organisms. They can function as enzymes. They can function as receptors for chemical signal transduction on the membranes of cells. They can function in regulating hormone activity. So proteins are very important molecules inside of the cell. Okay, they make up a huge portion of the cell. And again, the protein profile, expression profile at any moment in the cell is going to dictate how that cell functions overall. And so the goal of today's lesson is that you understand that in all of the cells, the thousands of cells that we have in our bodies, they all have the same DNA, they all have the same genome, but each cell gets their different function depending upon which proteins are created and expressed at any given time. This is just a reminder of the different options for protein structures that we've already talked about. But gene expression here, okay, is the key for regulating biological function. Okay, translation, all right, the synthesis of a polypeptide chain using the genetic code on a messenger RNA molecule as a guide. Okay, so that messenger RNA that we showed you earlier is the midpoint or a necessary intermediate that has to be made in order for protein synthesis to take place. So again, the concept that we're making here is that gene expression, okay, the expression of any particular gene, genes encode proteins. There are a variety of different cell types in our bodies. They all have the same genome, okay, but depending on which gene gets expressed and when determines the function of that cell. Okay, so cell function, gene expression is influencing development, cell differentiation, metabolic regulation. All of these factors are dependent upon gene expression. All right. We'll stop right here. This is a lot of information. I don't want to overload you, but I do want you to make sure that you understand this fundamental concept about the central dogma of molecular biology. The central dogma of molecular biology tells us how information flows from DNA to RNA to protein. You should be able to discuss, what this concept is and why is it important and how does it happen. We talked about the two processes of transcription and translation. Where do they occur in the cell? Transcription occurs in the nucleus. Translation occurs on ribosomes in the cytoplasm. For what purpose do we need transcription? Transcription is how we go from DNA to messenger RNA. Okay, translation is how we go from that messenger RNA into a final protein. You should understand simplistically how we use the genetic code to decode the information in our messenger RNA to create that linear sequence of amino acids. Okay, so we decode it using those codons. Okay, the codon was the sequence of 3 nucleotides. Each codon codes for an amino acid. And so once we get our line of amino acids, that linear sequence of amino acids, that becomes our polypeptide chain that ultimately becomes our protein. So I will stop this discussion right here and ask you, do you have any questions? Questions, comments, concerns?